Just trying to get my slides up, guys. What I'm going to be talking about today are practical aspects of microbiological method validation and verification. So I'm going to be talking about how you assure that the methods you use in your laboratory will give you the correct result. How do you actually do that? A little bit about me. I work for Campden BRI. For those of you who don't know Campden, we're a membership-based organisation based in the UK. So we've got about 2,500–3,000 members worldwide. We're an independent organisation and we really have clients in about 80 countries worldwide. I'm a microbiologist. I've worked at Campden for over 30 years, and a lot of my work has been based around microbiological methods and microbiological method validation to assure they actually work. First of all, moving into the presentation itself, the first question I think we have to ask is, what do we really want from a test method when we're testing things for microbiology? Clearly we want ease of use. We want it to be economical. We don't want to pay over the top for it. We want to make sure we get the right results at the right price. We need it to be relatively fast because some microbiological tests tend to take longer to get results, the traditional ones particularly. And often we want a higher throughput. We need to know that we're getting the right number of tests going through in a time. So those are some of the key thoughts when we think about doing a microbiological test. Of course, it has to give the right result. And we have to categorise what do we mean by the right result, but that means it has to detect things that are there and not detect things that are not there. And if we fail to get the correct result, we get a range of potential outcomes. We can get good food being wasted and thrown away, or we can get bad food being eaten and causing issues and food poisoning and all sorts of things. So the question is, how do we ensure that that's the thing that actually happens? How do we ensure that we get the right result? And there are a range of things that help us ensure we get the right result. And the first thing is we have to use a method that works. And we gain the confidence that methods actually work by using validated and verified methods. And that's what I'm going to be talking about for the rest of the day. Once you've got that method and you know it's the right method, it's validated, it's verified and it works, then you have to make sure you use it properly. And using it properly is the role of laboratory accreditation. And that's where labs become accredited through ISO 17025 and various other things. So use the right method, use it in the right way. Now, if we look at laboratory accreditation, if we look at ISO 17025, quality system in laboratories, and we look at what it says about validation and verification, in section 7.2, on this slide, it says you've got to select verification and validation of methods. So it tells you you've got to validate and verify the methods that you use. And then it goes on to say that laboratories shall use appropriate methods and procedures for all activities. And then it goes on to say that the laboratory shall verify that it can properly perform methods before introducing them. And it also goes on to say the laboratory shall validate non-standard methods and laboratory developed methods that are used outside their intended use. So 17025 is pushing you towards using validated and verified methods. So if we view those 17025 requirements in more detail, validation, you use validation, you've got to use standard methods like ISO and SEN methods, and those methods don't need to be validated as long as they're used unaltered and within their intended scope. That's what 17025 is saying. If you're using an ISO method properly, then that doesn't have to be validated. It's already taken to be valid. If you're using non-standard methods, laboratory developed methods, or standard methods outside of their scope, then validation is required. Verification, it says, is required for all methods. And it says there in 7.5.1.5 of 17025, the lab shall verify that it can properly perform methods before introducing them. Now that brings us into the thought process of, well, what do we mean by validation and verification? What do those terms mean? And the answer to that, the definition of validation and verification, comes in another ISO standard. And it's one that I'm going to be talking about quite a lot over the next few slides. And that's the ISO standard 16140. And this standard covers the validation of microbiological test methods. And part one of the standard is basically the dictionary of terms. It tells you what all these various terms mean. And for validation, it says, as shown on this slide, validation is the establishment of the performance characteristics of a method and provision of objective evidence that the performance requirements for a specified intended use are fulfilled. That's validation. Verification is the demonstration that a validated method functions in the user's hands. So they're two slightly different things. And if I try to make that more simple, validation, general terms, it may show you that an aeroplane can fly at the correct speed and height and go from Europe to the USA. Verification shows you that I can fly you there. So basically, you may be very happy to get an aeroplane flight to the USA, but would you be happy if I was in the cockpit? So that's the difference between validation and verification. One is generally showing a method works. The other, verification, shows it works in the user's hands, actually in your laboratory. So we said I was going to talk about ISO 16140, this standard of validation in more detail. That standard has a number of parts. Currently it has 6 parts, there's a 7th being produced at the moment, and they all cover different things. Now the bits that I'm really going to be talking about today are 16140 Part 2, which is the validation of alternative proprietary methods. So these are things, the kits that you buy from manufacturers and use. And I'm going to be talking about 16140 Part 3, which is the protocol for verification of methods in a single lab. So this is what labs will use to actually verify that in their hands the method actually works. So what about the need for validation in food microbiology? What does it mean to us? And I'm going to be talking in a very European sense now, because we'll move to the regulations, the food testing regulations in Europe. And they're covered with Commission Regulation 2073/2005, and that's on the microbiological criteria for foodstuffs. And in Article 5 of that regulation, it says that the analytical method in Annex 1 of this regulation, and those methods are the ones that it tells you you have to use for the foods you have to test, shall be applied as reference methods. And in general terms, in that annex, all the methods are ISO methods. That's all it says you can use. However, if you move on, it says that alternative methods can be used, but they've got to be validated against that reference method. And if a proprietary method, so if this is the kit that you've bought, then that method has to be certified by a third party in accordance with ISO 16140 or other similar protocols. So what it's saying is, in this regulation for testing in Europe, you have to use the ISO method, but if you want to use a proprietary kit, you can, but that kit has to be validated against ISO 16140 Part 2. Has to be validated. Now, interestingly, there are some proposed changes to that 2073/2005 regulation that are being proposed at the moment. Now, these aren't actually there yet, but they're being proposed, and on this slide you can see what those actually are. And it changes some bits and pieces. And if we go through this and look at the red figures, let me just see if I can put a pointer on that. And we look at these red figures here. It says that the alternative methods have to be validated in accordance with 16140 Part 2, validation of proprietary methods. They've got to be validated for particular food categories. So that's another part. They've got to be valid for the food you're going to test. They've got to be set out and validated against 16140 Part 2, as I said beforehand. You can also use alternative confirmation tests if they're validated against 16140. But the key part of these proposed new statements that are coming in is what you can see down there in 5G right at the bottom. And it says the use of a reference method or an alternative method, so either, can be used provided the laboratory can demonstrate the verification of the performance of that method as set out in the protocol of 16140 Part 3. So this is now making it very clear that labs have to verify in their own hands that methods, whether they be ISO methods or proprietary methods, work in their hands. It has to be done, and it has to be done according to ISO 16140 Part 3. As I say, those aren't in the regulation yet, but they're being proposed at the moment, so something to look out for. Okay, if we move on now and look a little bit more at one of those standards, and that's the Part 2 standard, which is the standard that covers the validation of proprietary test methods. So these are the kits that you buy off the shelf from manufacturers to do particular types of test. Now, the way the validation is done shouldn't really be a concern of individual laboratories that are using the methods. The validation is actually done by an expert laboratory. It's done once by one expert lab. And it's managed by a validation organisation. And there's a whole range of validation organisations. There's Microval, AFNOR, Nordval, AOAC. They're all validation organisations that will validate and certify methods using the ISO 16140 protocol. As I say, it's not done by you, the user laboratory, it's done by the expert laboratory. So what is 16140 Part 2? And I've put Part 6 in this because Part 6 is basically how you validate confirmatory test methods. 16140 Part 2 is looking at how to validate testing methods and confirmation methods, and it covers quantitative and qualitative methods. It's done in two stages. There's what's called a methods comparison study, and that methods comparison study is done by the expert laboratory in their lab. And it's quite an extensive series of tests. Campden are an expert laboratory. We do a lot of work for Microval and the AOAC, and it's quite an extensive series of tests that you have to do to compare the new method, the alternative method, against the reference method to prove that you're getting equivalent results. Second part of 16140 is the interlaboratory study. And in this part, the expert lab will send samples of a food out to between 12 and 15 different labs, and they will have to use the alternative test and the reference method to prove that they can get the right results as well. And it's really showing the robustness of the test method and the variability you might get in other laboratories. So that validation generates key performance characteristics of the method, and the output indicates if a new method gives results that are at least equivalent to the reference method, the ISO standard method in this case. So one of the questions you may raise in quite a valid way is, how do I find out if a method is validated and certified? Very easy to do. And on this slide, I'll giving you the websites of all of the major certification bodies. And basically, if you go on those websites, you will be able to find lists of validated methods, and you can find the details of the validation. So that just gives a quick snapshot of the Microval website, and it gives you a range of test methods. And if you click on the little blue number there, you will go into the certificate and the test report that shows the validation. So you can find it out relatively easily. So, taking a step back, it's easy to find a suitable method by looking at these websites. You can look for methods that are validated and certified by these certification bodies to ISO 16140 Part 2, and therefore meet the requirements of European regulations. So basically, you might think, I'll just do that, I can take that method, I can use it, it's fine, isn't it? Well, then we have to ask the question, is it fine? Because 16140 actually says the selection of categories and types of food used within the validation will depend on the type and group of organisations and the scope of the validation. Have to remember that the validation itself has a scope. And within that scope, the laboratory, the expert lab, may test lots of food types or very limited numbers of food types. And that's something you have to be aware of when you're picking a method. Does the validation cover the food that I'm going to test? Let's take an example. What do I want? I'm working in a lab. What do I want? Let's say I want a validated fast testing method for E. coli 0157 to use on my soft cheese. Don't want to use the reference method, takes too long. I want to use a proprietary method. I want to look for one that fits my lab needs, that has the right costs, that's been validated. That's the job done, isn't it? Let's take a closer look. Let's take a look at this. This is an actual AFNOR certification letter. And basically, you have to look at this in some detail to decide if this covers what you want. So remember, I want to test my soft cheese for E. coli 0157. Let's look at that letter in a little more detail. Right, things you need to look for, listed at the bottom of this slide. Date of validity. Is the certificate still valid? And if we look at the date of validity up there, it says the 2nd of July 2022. So at the moment, that's still valid. Secondly, reference method. Is the reference method correct? And if we look here, the reference method that's been used is ISO 16654, which is the reference method for testing for 0157. Reference method's okay. Then look at the scope. Foods. Is it acceptable for you? And if we look here, it's been validated for detection of 0157 in raw beef and raw vegetables. What do I want to test it for? What do I want to test? I want to test soft cheese. This method has not been validated for the food type I want to test, therefore I can't use it. It's not a valid method to use. I have to look somewhere else. Let's move again. Okay, this is a microbiological certificate. And let's say in this case, I want to test environmental samples for Listeria monocytogenes. I'm doing environmental testing. So let's look again. We've got the date. The date there said the expiry date is the 21st of November 2022. Date's fine. Reference method, somewhere in there the reference method will be listed, and it's 12290 part 1, which is Listeria monocytogenes ISO method. Fine. Organism is fine, it's Listeria monocytogenes, it does what I want it to do. Let's look at the scope again, the sample types. It says the scope here is raw milk and dairy products, heat processed milk and dairy products, ready to eat and reheat meat products, and ready to eat and reheat fishery products. I want to do environmental testing. It's not in the scope. So really beware that you can't just pick up one of these validated methods and say it's validated for Salmonella or Listeria and use it without looking in detail at the certificate. Does it cover the food types that I want to be able to test? So what's it mean? It means you have to check validated methods really carefully as a user. Is the certificate valid? Check the dates. Has it been done with the correct reference method? Is it the ISO method that I want to use, want to use against? Has the validation scope covered the foods that you want to test? Is it valid to be used on the foods you want to test? Does the certificate cover the sample mass that you want to test? It's really important because as we look at some methods, particularly for things like 0157, STEC and Salmonella, we may see the sample weights increasing. Maybe people that want us to test 375 grams, not 25 grams. If it's only been validated for 25 grams, it's not valid for use with 375 gram samples. You can test with lower sample masses, not higher sample masses. Another good one to check. And does the certification cover the organisms that I want to test for? Really key things to think about. So, does that mean we're now good to go? Are we okay now to go and use that method? Well, no, actually we're not. Because the method's been validated and it's been certified. So basically, in my simple terms, my plane flies and it covers the distance, but would you want me to fly it? Verification is now required. And this is where individual laboratories come in. Every lab has to verify that the method that they've picked can be used in their laboratory and get the correct results. So, how do we go about verification? The way we do it is quite interesting. In the past, if you look at ISO 17025, which is the laboratory quality system standard, it doesn't really tell us anything about how to verify methods. It tells us we have to do it, doesn't tell us how. So up to now, labs have tended to do it very differently. Some labs have done very extensive verifications, others have done far less. And it's up to the laboratory accreditation organisation then to look at the results of those verifications and decide whether it's acceptable to that laboratory accreditation body. There's no standard. And the result of that is that verification has been done in what I would say is a very non-standard and quite haphazard way. Different labs have done different things, and some of those have been found acceptable by different bodies in different countries. Complete lack of standardisation, and that's led to a degree of confusion. And certainly we've had a number of people coming to us saying, well, how do I verify? How much work do I do to verify my method in my laboratory? And it's very difficult to give them an answer. So that's what has been the procedure in the past. But as of the late part of last year, 2021, we've seen published the standard ISO 16140 Part 3. Now 16140 Part 3 is a part of the 16140 series of standards, which is for a protocol for the verification of reference and validated alternative methods implemented in a single laboratory. And basically what this standard does is it sets out an international approach to ensure consistency to laboratory method verification for reference methods and validated alternative methods. This is a key part actually, because where we've been talking about method validation, and the only methods you need to validate are proprietary methods. ISO reference methods don't need to be validated, they're taken to be valid. Methods that have to be validated are proprietary methods. When we move to verification, so this is the methods that you use in your lab, it doesn't matter whether you're using an ISO reference method or whether you're using a proprietary method, they have to be verified. All of them do. You have to do this in your own laboratory. So this is what 16140 Part 3 is doing. And what it does, it standardises how to do verification. So it's talking about the approaches, if you need to spike and the spike levels. It tells you how much work you need to do, so the number of food items you need to test, the number of replicates you need to do. And it tells you how to appraise the results. So it's really important, because beforehand we had no guidance at all on how to appraise the results of verification. Now this standard tells us how to appraise the results. What means a method that you're using passes verification and what means it fails. So it's an important standard as we move forwards, but of course it does mean that laboratories are going to have to start doing maybe a little bit more work in verifying that the methods actually work in their hands. So let's look at ISO 16140 Part 3 in more detail. So we're talking about method verification, and that verification proceeds in two stages. The first stage is implementation verification, and the second stage is food item verification. And this is what you would have to do in your laboratory if you were verifying a method. So let's take those two stages and describe a little bit more about what they are. So implementation verification. This basically is showing that your lab can run the method correctly by getting results that are similar to those obtained in the validation study by the expert lab. So it's comparing results that you get in your laboratory with results that were obtained by the expert laboratory during the validation process. So it's really, it's comparing results between you and the expert lab to show they're approximately the same. So that's giving you the first part, implementation verification. The second part is food item verification. Now, food item verification is very much linked to your laboratory, and it's demonstrating that your laboratory can run the method with the food items that you want to claim your laboratory can test. So it's really aimed at what you actually test. And this is where the work can be small or large. If your laboratory is a lab that's only testing let's say milk samples, then of course you'd only have to verify milk samples when you do this work. If you're a contract testing laboratory and you have a wide range of different sample types coming into you from different clients, then you're going to have to verify many different food types to show that your lab can actually get the correct results from them. So food item verification, the amount of work needed might be small or large depending on the type of samples you're actually testing. So before performing the work, before performing these two types of verification, what does the lab need to do? You need to be able to refer, first of all, to either reference methods that quote performance data for the foods that you're going to test. Now, if you start to look at the new, the newly published ISO standards, and they're being republished as time goes on, the newer ones, when you go to the back, will have a section on performance data. Now that performance data is the validation data for that ISO method, and that's key when it comes to implementation verification. If you're using an ISO method, you will be looking at that back part of the ISO standard, and that will be your comparator data when you're doing implementation verification. Moving to that second bullet at the bottom, if you're using a proprietary method, a kit that you've bought from a company, then what you'll be looking at are those validation reports, the things that we've talked about earlier, the AFNOR, the Microval, the Nordval and so on information. You'll be looking at those validation reports, and they will quote the performance data for the foods that they've tested, and that's what you'll be using as your comparator against the information, the results that you get in food in implementation verification. So that's where to start from, and it's key that you need to have that information on performance data from the comparator method before you start doing the verification that you're going to do. So what do we measure when we're doing verification? Well, it depends. Depends on whether you've got a qualitative test or a quantitative test. And that's shown on this slide. And if you look at the table at the top, with qualitative methods, so presence-absence testing for salmonella or listeria or whatever, then the performance characteristic that will be being considered is the estimated LOD50. Now the LOD50, as we say at the bottom of the slide, is an estimate of the level of detection at 50% probability based on an experimental design. So it's what level do we start to only detect 50% of positives? So that's what we'll be trying to measure. It's really measuring the limit of detection of the test. How low can you go when still detect the presence of the organism? And that use of the LOD50, the estimated LOD50, will be the performance characteristic that you used for both implementation verification, that's your comparison against the expert lab results, and for food item verification. You use the same characteristic for both. If we now move to quantitative methods, so these are your counting methods, your total viable counts, your Enterobacteriaceae counts, your E. coli counts, and so on, then it's slightly different. Because you'll be using two different performance characteristics for both implementation and food item verification. For quantitative method implementation verification, so this is your comparison with the performance characteristics obtained by the expert laboratory, we use something called Interlaboratory Reproducibility Standard Deviation. It's a nice statistical term. I'm not a statistician. I don't particularly understand it. Doesn't matter, because the ISO standard has links to programs that allow you to put the data you generate in, and they'll do the calculation for you, which is great for non-statisticians like me. So you use this SIR value, this intralaboratory reproducibility, for doing implementation verification characteristics. When we move towards food item verification, so this is looking at verifying that you can test the range of foods that you use in your lab, we're using a characteristic called bias, the estimated bias, and that's what we'll be looking at. So there tells you at the bottom there, the bias is basically an estimate of the systematic measurement error or difference between the assigned value and the average of your measurements. So the assigned value is the real value, basically, of the number of microorganisms, as opposed to the value that you get doing your test. So let's have a look in a little more detail at how you move forwards and do verification. So first of all, let's look at the verification of qualitative methods. And let's look first of all at the implementation verification. So this is the comparison of your results with the results that are obtained in the validation study by the expert laboratory. And what we're looking for here is this estimated LOD 50, the point at which the method only detects 50% of the positives. Now, to do this, you only need to test one food item. Great relief by everybody. And the food item you're going to test is any food item that was used in the validation study by the expert laboratory, that's within your lab scope. So you must be able to test it as well. However, if you find that the validation didn't use a food that was in your scope, that's fine. You're just going to have to pick one of the foods that was used in the validation study and use that, even if you don't normally test it. It's key that the sample size that you use in your lab has to be exactly the same, or it can be smaller, than the sample size that was used in the validation. You can't go bigger. You can't pick a bigger sample mass. It's got to be the same as the sample size that we used in the validation study. Now, when it comes to doing the actual work, the standard 16140 Part 3 actually gives you 3 different protocols that you can use to do implementation verification of qualitative methods. And the choice, and you can choose any one, the choice depends upon the ability of the lab to achieve a desired level of contamination, because you're going to have to contaminate these food samples using a dilution of the target organism. And it gives you 2 protocols where you inoculate the foods at varying levels, and it gives you 1 protocol that requires the use of reference material when the level is actually known, and that's a reference material you would buy from a company with a known level of microorganisms. Now, the key thing, and it's the third hash line coming down there that's on my laser pointer there, is you're going to use this method for both implementation and food item verification. Same procedure. But you've got to use 3 protocols there. You've got to use the same protocol for implementation verification and food item verification. You can't change from one protocol to the other. It has to be the same. And it requires the artificial contamination of the food sample you pick. Now, approximate amount of work that you need to do. The total number of replicates you'll need to do is about 8 to 10 for the one food item. So it's not an enormous amount of work you're going to have to do. But the key thing is getting the inoculation level right, and that may take a little bit of time because many labs aren't used to inoculating foods with particular low levels of microorganism, so it may take a little bit of time to get that right. The standard gives you examples of how to do it. As I said before, the approach to food item verification is exactly the same as that for implementation verification. It's the ELOD 50 route, and what you're going to do is be inoculating the foods that you use in your lab in the same way and getting results. And then the standard will give you a way of assessing whether the results that you get meet the requirements, whether it's been verified or not. Okay, let's move now from qualitative methods, presence-absence tests, to quantitative methods, so counting tests. So how are we going to move forwards with this? I said, you're going to use a different procedure for implementation verification, the comparison of your results with the results obtained with the expert lab, and food item verification, what you use in your laboratory. So let's look first of all at implementation verification, and a key comparator, the key performance characteristic we're going to go for is the SIR value. This is the intralaboratory reproducibility. And the way that's done is you first of all select any food item that belongs within the scope of validation of the laboratory. Best to choose one that can be effectively homogenized, because we're going to be doing inoculation here, and to get a good homogeneous mix, pick something that you can easily homogenize. You know, a sandwich may not be the food you want to go for. A yogurt or milk or something that can be easily homogenized might be. If possible, best to use a naturally contaminated food, but we all know how difficult it can be to find naturally contaminated foods with some things like E. coli or Staph aureus and so on. Because the final point there said, if you can't get a naturally contaminated food, then you can artificially contaminate it at relatively low levels. So moving forwards, how do you then do the testing work? How do you measure this intralaboratory reproducibility, the SIR value? Well, actually, it's quite fortunate in some ways that the protocol that's detailed in ISO 16140 Part 3 is the same protocol that's used in another ISO standard, and that's this 19036, which is the way a laboratory would measure uncertainty of measurement. So if your lab has used the ISO 19036 to measure uncertainty of measurement, it's exactly the same protocol that's going to be used now to do intralaboratory reproducibility for method verification. Basically, you will pick a food item. You need a minimum of 10 laboratory samples of the same food item, and this is trying to get samples that you could see would be varied. So it's saying samples of the same food, maybe from different batches or maybe from different manufacturers. So try and introduce some variability in the samples you use. Same sample type, but some variability in them. You've got to pick, you've got to obtain a minimum of 2 test portions per each of those samples, and we then run the protocol. And this, I'm not going to go into great detail into that flow diagram, but essentially you have to inoculate homogenized food samples with the test microorganism. You homogenize the food before you take the test portions forwards. You spike after homogenization to get a good mix of the microorganism in the foods. You then have to vary the parameters across the different test portions of each food sample type. By doing that, we're trying to get intralaboratory differences. So what you're trying to do with the variety, with the variation you're doing, is use different analysts. That's variety. Use different pieces of equipment for each of the different test portions, different incubators, different pipettes. So we're trying to deliberately produce variations in the way you do the work. Once we've done that, we've got results. And again, as I say, I'm not a statistician. Fortunately, the standard gives you a link to a program that you can simply input the results that you get, and it will calculate this intralaboratory reproducibility standard deviation for you. And then you will compare it to the requirements of the standard, see where it meets requirements or not. I think we got it on the next slide actually. What we're looking at, the acceptability limit that's in the standard says that the reproducibility of the verified method should be less than or equal to twice the lowest mean value that was found in the validation study. So if you look at the validation study reports, either in the ISO standard or in the AFNOR or Microval certificate, then it's allowing you a little bit of room for manoeuvre, and it says it's acceptable if it's twice that particular value. It's giving you a little bit of room for manoeuvre there. And if it is equal to or less than twice that value, you've verified it. You've done implementation verification of that method in your laboratory. And then you can move forwards to do food item verification for that method. Now, we said beforehand, we're using a different approach here for food item verification. We're using something called e-bias, the estimated bias. You take the food samples that you use in your laboratory, and you artificially contaminate them at 3 different inoculation levels. And then you test an uninoculated portion to make sure it's not naturally contaminated. And then you enumerate each of those inoculated test portions that you've produced. Your comparator is the number of organisms that was in the suspension that you used to inoculate the samples. So that's your comparator. So what you're doing is comparing the results that you've got from using the test method of inoculated foods back to how much did I actually put in the foods to start with. Okay, e-bias. How do we look at the results? Results evaluation. You compare the results from the artificially contaminated foods to the results of what you inoculated in. And the acceptability limit that's noted in the standard, which is that second bullet point there, is the absolute difference between the 2 results should be equal to or less than half a log. So you can have half a log difference between the culture that you inoculated when you enumerate it and what you actually enumerate when you've inoculated into the food. If you've got less than half a log variation, you've verified that you can use it for that food. And you simply have to do that then for the number of foods you've actually got. Now, one of the questions that's raised that is real importance at the moment is, so how would I verify something if I had a non-validated ISO method? If I was using the ISO method for the work I'm doing, then I'm a bit stuck because the validation data has been done centrally and it's all written up in the back of the ISO standard. And as I said beforehand, the standards are gradually being republished with this data in the back. There's an awful lot of them that don't have that validation data in the back of the ISO standards. So what happens if I'm trying to validate a method to use in my laboratory if it's not been validated and the data doesn't appear in the ISO standard? Well, what 16140 Part 3 actually says is, in that case, you only have to do food item verification, and you use those approaches of ELOD 50, reproducibility standard deviation or e-bias. So you don't have to do implementation verification. You can't do it because you haven't got the data to do it. You only have to do food item verification. However, that will only last until this date noted here, 31st of December 2027. And then all methods should be validated. Now, this is putting a great deal of pressure on the ISO system, because what it's basically saying is at that point, ISO have got to make sure that all of their test methods contain validation data in the back of the standard, because that's what you've got to use then to verify. And there have been various, this isn't included any of the standards, but this is a piece of information that's been widely given out by ISO and other parties. There is a transition period when laboratories actually have to start doing verification according to 16140 Part 3. And what it's basically saying is, currently, if you have methods that you have already got accredited and they're in your scope of accreditation, you don't need to re-verify those methods using the 16140 Part 3 approach unless you make changes to the method. So if you've already got those methods included in your scope, you've already verified them using whatever method you've used beforehand, it's been accepted by your validation body, then that's fine. You don't need to verify those using the 16140 Part 3 method, unless you make a change. If you're going to change the scope of application, so if you're going to add a different food type into what you're using with your method, then you're going to have to verify use according to 16140 Part 3. So anything you add in, if you change the method in any way, if you revise the method in any way, if you bring a new method in to be used in your laboratory to add to the scope of your accreditation, then any of those changes you're going to have to verify using ISO 16140 Part 3 methods. So, in some summing up, some final thoughts what I'm going to say. We've come a huge way in ensuring that the quality of the methods that we use in our laboratories, our microbiological testing laboratories, come an enormous way. In the years I've worked in microbiology, we've stepped forward an enormous amount, and we've got much more confidence that the methods we're using are being used properly and give the correct results. We now have some very well understood and well used methods for the validation of new methods for proprietary test methods, and an awful lot of methods that have gone through those validations. If you look at the lists that are in the AFNOR and the Microval websites, enormous, and AOAC. Most actually proprietary methods, kits that you buy, are now being properly validated before being marketed. That's going to give us great confidence that those methods are giving results that are fit for purpose. Your job, when you start to think about picking a new method, is to make sure that those validations cover the foods that you're going to test and the way you're going to use the method in your laboratory. Check the scope of the validation to make sure it's fit for the purpose that you want to put the method to. Check them very carefully, check the dates, check the reference methods, check the food types and how they're going to use it. Make sure they fit. Standardized verification in your laboratories to prove that your methods actually work and give the correct results is really at quite an early stage. It's always been done, but it's been done in this plethora of different ways. We're now just beginning to get that standardized approach using the ISO 16140 Part 3 standard. It may look very complex. If you're not used to this, if you've not read the standard, it may look very complex at first when I go through it. But in the fullness of time, all laboratories are going to have to use it. And I think once labs start to use it, they'll realise that it's actually a lot simpler than they at first thought. And the benefit of doing all of this is we will gain much better quality of the results that we get and a much better confidence in the results that we get, so that we're confident that the results actually do refer to the actual microbiological nature of the foods that we're testing. Thank you very much.
Thanks very much, Roy. We'll now engage in a short Q&A session, if that's okay.
It's fine.
So obviously this is open to all of the attendees. If you wish to ask a question, just use the chat box functionality. It's on the right-hand side of the screen. And during the presentations, don't forget to answer the polls that Julian is putting up as well. It's on the right-hand side of the screen, and it's the little icon with the 3 columns in it. So, first question from Julian, as it happens. Typically, what kind of resources in terms of people and time are needed to perform an ISO 16140 Part 3 verification in a given laboratory?
Okay. I think it would be wrong to say that you don't need to put some time into it. We've actually this year gone through and verified a number of methods in our laboratory. We tend to use a lot of ISO standard methods for the work that we do. And we've probably gone through we've probably verified somewhere in the region of 7 or 8 methods. In man time, that would probably be about, what would it be?
A month and a half?
I would say a man month
yep.
of time in order to do that put aside. So think of a man month, 2 people for half a month, whatever, yep. in order to do it. You only have to do it once, but it's time that has to be put aside to do that, yes.
Yep. Okay. And also in the chat box, my colleague Verena has put a link to a survey to be filled as well. Okay. We have another question from Gift. Is there a requirement to re-verify methods periodically, say every 2 years, even where they've not been any changes?
I don't believe that the standard actually says you have to re-verify unless you change the method.
Okay.
Or, well, let me go through it. If you change the method in any way, if you include more foods into the scope of validation, and I would probably say it will be good practice if you changed any key part of the kit that you use, or maybe the laboratory. So if people moved laboratories, so if they built a new lab and they moved everything out, then you probably want to do some work to re-verify some of the methods to show that things work.
In that different environment.
Yeah, different environment. So, you know, if you've got a new piece of kit, if you're doing PCR and you've got a new thermocycler, or if you know, even a new incubator change and things like that, you may want to just do some work to prove that the verification's still held. But I don't believe within the standard it says that you need to constantly re-verify methods at a set point. I think for many laboratories, if you hand on heart really wanted to prove that things were continuing, you may want to pick different methods at different times and just do a quick re-verification to make sure you were still happy things were going okay.
Sounds logical. Another question from Angela this time. Whilst performing validation and verification, is it worth sending out representative samples to another laboratory to ensure you're attaining similar results?
With validation, which is the one that's done by the expert lab, that's sort of done anyway in the ring trial, because the ring trial then is coping with what happens in other laboratories. With verification, no. I think as a lab, you will have other ways of assuring you're getting comparability with other labs, because you should be doing external quality assurance checks. So you should be getting samples coming in from an EQA provider, which compares your results to everybody else's results. So you should be getting that comparability anyway. Within the verification, no, that's completely internal to your laboratory.
Okay. Any questions directed at Roy from David or Han?
Yeah, maybe one question about these different food categories, because I don't know what guideline or I understand.
Good point, a very good point, yes. It really is a good point. Actually, I took a slide out of that yesterday, but maybe I should have kept it in. And I don't think I've got it on there, no. Within ISO 16140 Part 2, which is the validation standard for validating proprietary methods, it has a table of food category types. And I think 12, there are 15 different food category types that it states, and 3 additional categories which cover environmental samples, primary food samples, and something else I can't remember.
Environmental.
Yeah. So that gives you these 15 categories that will be tested within the validation. Now, you can pick, within validations, you can pick which of those you test. So some of those that I was showing had limited scope of validation, so then you picked 1 or 2 of those to test. So, you know, maybe for you doing 0157, maybe you may only want to validate for beef, because that's what most people do. If you were doing salmonella, maybe you'd have a much wider. But within 16140, if you test 5 of those food categories, then it will be validated and certified for what's called a broad range of foods. So that means really it's accepted at the validation level as being valid to test anything with. That's where verification becomes really important, because you may be testing something that's not been validated in your laboratory, and that's where verification becomes very important. So there is this table within 16140 of these 15 key food categories, and that's what the validation laboratory will always validate against. The food category is there, and you can find it in 16140 Part 2.
Now, the reason for asking is also because, particularly for presence-absence methods, you may have food categories that perhaps sound like infant formula, But what will you do if it also contains probiotic bacteria?
Yes, yeah.
That is probably not covered. That would be too much work.
Oh, too much.
But you have to use common sense.
And this is, I think, the real importance of the verification part, because it's impossible to validate for every food type. All the validation is doing is giving people a general comfort the method works. When it comes to verification, that's when that becomes important, because that's when a lab will start to be using the samples it knows, hopefully it knows, and those issues of probiotics or antimicrobials or whatever in the foods really become important. And that's what verification will show.
Perhaps a quick question from an attendee for you to answer. Does this apply when validating culture media? So I think they're referring to verification process.
The verification. Okay, let's go through it. It's an interesting question. Culture media. If the culture media is being used as an alternative to media within the ISO standard, and some are, it probably needs validation. Okay. Using 16140 Part 2. And if you look at some of the 16140 Part 2 certificates, there's a range of culture media that have been validated 16140 Part 2. Alternative agars, alternative broths, and you'll find that there. That has to be validated to prove it gives equivalent results to the reference method. Okay. If you have made a change, if you're using an ISO method standard, okay, so it results are in the ISO standard, and you make a change to something, so I don't know, different incubation time, different incubation temperature, you are going to have to verify that that's fit for purpose in your laboratory. You may even have to do an intralaboratory validation of that. So you've got to be quite careful. If you're making changes in your laboratory, that might have a really big implication to you having to validate, not verify, validate that that method can be used.
Yep. Okay. Another question. What possibility do I have if my food item is not in the scope of the alternative method, but I want to verify it?
Okay. I'm just thinking about validation and verification. So if the food item isn't included, read that out again.
Yeah, sure. What possibility do I have if my food item is not in the scope of the alternative method, but I want to verify it in lab?
That's a really difficult one. So it's not in the scope of the method. We're not talking about validation now, we're talking about the scope of the method. It becomes really difficult because a method manufacturer is going to say, my method works with these things. And the validation will go, well, you've said that, I'm going to validate whether it works or not. And that's what the validation is. If you're talking about using a method where the method manufacturer says, my method isn't valid to be used on that, that's taking a real step into the dark. You would then have to do a full validation to prove that that method is, which is outside of the scope of what the manufacturer says you can do, you can actually use. If it's simply something that the method manufacturer says it works but it hasn't been validated, then you then have to validate to that actually works. If it's included in the validation, but it isn't, if it's a broad range of foods, but that food isn't included in a broad range, you simply have to verify it. So it depends exactly what's in the scope of the method, the validation and the verification. They all have different scopes. It's quite a difficult question to answer, that one.
Yeah. Let's see. Perhaps the last question, looking at the time. Back to the culture media.
Yeah.
What if it's just a change of brand due to unavailability of the normal brand?
Really good question. We got caught by this. That really depends on a number of things. If the recipe of the different brand is the same as the one you're using, then there shouldn't be an issue. You may want to do, for your own benefit, you may want to do a quick test of one against the other to make sure it's okay. Shouldn't make any difference. What can catch you out and I'll be honest, it's caught us out in the past, is if your laboratory accreditation says that you're going to use a particular brand of media. So, as I say, we've had things in the past where we've stated in our method that we will use manufacturer A's buffered peptone water. And then manufacturer A goes, it's short, we can't supply it to you. We go, okay, we'll use manufacturer B, it's the same recipe, but we've stated in our method we're using manufacturer A. That then becomes a real issue with laboratory accreditation, because the method says we'll use A, we can't simply switch to B. If within your method you simply say, I'm going to use buffered peptone water of ISO recipe, you can use whichever you like.
Yep. So the headline would be check your method. Check
your method and maybe don't make it too specific.
Okay. Wonderful. Thank you very much, Roy.
Okay, thank you.